Okay, resuming for the second part. So we are talking about the adaptive immune system. We talked about how the stimulation takes place through the presence of receptors, the B cell receptor or the T cell receptors. And then we talked specifically about the activation of the antibody-mediated immunity. Now we're going to talk about the second type of adaptive immunity, which is the cell-mediated immunity. In this case, what is happening is that the pathogen, so the antigen present in the antigen, so it's going to be presented to a different type of T cells. So if you remember, you have the antigen presenting cells, so these dendritic cells that have engulfed the pathogen and broke it down into different antigens that the first have presented to CD4 plus T cells through the major histocompatibility complex 2, and these T cells become helper cells. Now the same cells can actually activate also a different type of T cells, which are referred to as CD8 plus cells, T cells. So these cells instead, they get activated. So the antigens for these cells is presented through a different major histocompatibility complex, which is a class 1 major histocompatibility complex. And these cells, when they get activated, again, through their T cell receptors, through the production of interleukins as well, in this case, they don't convert into helper cells, which then will stimulate the B cells, but they actually get activated into effector T cell or cytotoxic T cells that do directly will act against the pathogen. Okay, so this is what is shown here. Now you have the antigen presenting cell here, use a different major histocompatibility complex, type 1 in this case, or class 1, to present the antigen to the CD8 plus, T cells. Now these cells, they don't turn into T cells, but through interleukins here, they get stimulated to expand to turn into cytotoxic T cells, which then will go and produce molecules, perforins, and different type of other molecules, which actually do kill the cells that have been infected by the virus or the bacteria, whatever it was producing that. So this is how cell-mediated is produced. Cell-mediated response is produced. So this schematic here shows you the two responses. Again, the antibody-mediated response and the cell-mediated response, which are both part of the adaptive immunity. So in antibody-mediated cells, you have that the antigen-presenting cells presents, first of all, the antigen through a class 2 major histocompatibility complex to first CD4 T cells, which then get stimulated by interleukins to expand and become helper cells. So CD4 plus T cells become helper cells, which then will stimulate, they bring the antigen basically to the B cells, because now they all have this receptor, T cell receptor here, which is specific for that antigen. They bring it to the B cells, which then they would expand clonally. And it will lead to the formation of plasma cells producing the antibodies and some memory cells there. Now in the cytotoxic or cell-mediated response, what you have is that the antigen presenting cells now use a different type of major histocompatibility complex of type 1 to a different type of T cells. So these are CD8 plus cells, which then they get expanded through the production of interleukin, and now they do act directly on the infected cells instead of presenting the antigen to the receptor, to the B cells. Now, an important thing is I forgot to mention, all of this usually happens in the lymph nodes. That's one of the reasons why sometimes you may have the enlargement of one of your lymph nodes. It's usually a scary thing when it happens. You never know if it's cancer, you have a lymphoma or something like that, or oftentimes is due to exactly a very active infections. You have that, you have an antigen that's stimulated, overstimulated your B cells and T cells to expand there, and therefore you get a lump on that lymph node. So that's an example. But other part of the lymphatic tissue can also be parts where these happen. Now, On the side here, you can see how these antibodies here, now that they are going to be produced by the plasma cells in the antibody-mediated cell response, now you have a series of antibodies that are very specific for that antigen here. So this is great for your immune system, but it's also great in research as well. So if you can actually do that, you can actually stimulate some animals by exactly presenting them a certain antigen. You can stimulate them to basically stimulate their B cells to produce specific antibodies, which then you can extract and use in research to detect specific antigens that you had first injected. So these are referred to as monoclonal antibodies, are all antibodies that are specific for the same antigen and particularly epitope. So, and they can be used, for example, to visualize a certain structure inside a cell. So imagine you take the IGF-2 receptor and you inject it into rabbits. Now you can get the rabbits to produce antibodies specifically against this protein, which then you can take out, purify, take some cells, treat them with the antibody. This antibody will bind only where the IGF-2 receptor is. And then you can make that visible by using a second antibody, which is labeled with a fluorescent molecule on the constant part. And then this basically will allow you to visualize where this receptor is found. This can be used for many other things. It can be used for cancer research as well, and so on, cancer treatment, and so on. Now, sometimes, this is a perfect system that I just showed you, where you have an organism that enters the body, some cells, they ingest it, produce antigens, which then stimulate the cell-mediated and the B-cell mediated or antibody-mediated response. This is the perfect system. Sometimes these things do not happen. Some organisms, they do actually manage to escape that. They do not stimulate this system. There are different ways that this can happen. Let's move on faster on this part right here. Sometimes you have autoimmune, so I'm not going to talk about that. But what I want to give as an example is how HIV, for example, managed to escape your immune response. So you can see how it fits into the mechanism I just described. So you can have different ways that organism can escape that. It can, because simply they invade regions where your B cells or T cells cannot go, and so on. But for example, when it comes to the HIV virus, that's how it escapes. HIV virus, it infects specifically CD4 T cells. Do you remember, these are the cells that then will become helper cells in the antibody-mediated immune response. So how they do that? They do that through this protein called GP120, which then binds the CD4 receptor. That's the one that normally will bind the antigen, right? When they do that, they manage to enter inside the T cell. And there, what they do, they actually integrate themselves. These are RNA viruses, but they do produce DNA, which then integrates inside the T cells DNA. And now what happens is that also, if you remember, this HIV is named HIV because it stands for human immunodeficiency viruses, meaning that also it causes immunodeficiency other than the infection itself. How is that caused? Is that because once it's integrated inside the DNA, now when a cell, one of these T cells, right, that now would have the virus inside, gets activated to respond against a certain specific antigen. Now this cell will be stimulated by interleukins, all normal here. But when it gets stimulated to grow, now the virus will start actually growing too. It's integrated into the DNA, it will start being produced in high scale. And so the cells that are actually expanding clonally here, at the same time, they are producing so much virus that then they die. So what you have is that not only you have the virus reproducing itself, but at the same time, it's actually shutting down the helper T cells once they start reproducing, and therefore they cannot stimulate anymore the B cells, which they're supposed to stimulate. So the virus is replicating, at the same time it causes immunodeficiency against many other type of infections as well. Last thing I'm going to discuss here, and then we're done for the whole semester, is how that variability of antibody is generated. You have so many different B cells in your body, which are normally produced even in the absence of any antigen entering your body. You have so many B cells ready to act against so many types of antigens. That's how they're made. But if you think, all of these B cells to start with, they have the exact same genome, right? And now what you have is that, if you remember, what they're stimulated with is through the B cell receptor. This works for the T cell receptor, but in this example here, what I'm talking about here, I'll focus only on the B cells, okay? But now, if you look at the B cell receptor, which is what starts all the response there, is produced through basically 3 genes, okay? So you have, I will talk about actually in the next step. So there are 2 ways in which the variability is produced. Through the first step, which is referred to as somatic recombination or VDJ recombination. And then you have the somatic hypermutation. This is done after the stimulation of the B cell receptors, which fine-tunes basically the antibodies produced. So before we talk about how the variability is produced, so this is a schematic of how the B cells mature in the bone marrow first. So you have, they're all generated from the hematopoietic stem cells, which first produce lymphoid stem cells, which then produce the progenitor for the B cells and the T cells. The T cells, the progenitor T cells here will move to the thymus. That's where it will mature into a T cell. The progenitor B cell here now will go 2 more steps of maturation. In the first step, they become a precursor B cell. And here you have the first type of variability generated through the heavy chain arrangement. You'll see in a second what I mean. And then when it changes from precursor to immature B cells, which then will move to the lymph nodes later on, there's a second level here of variability, which is the light chain rearrangement. If you remember what we're talking about, heavy chain and light chain, we're talking about the peptides that make up the B cell receptor. Each B cell receptor has 2 heavy chains identical to each other with a constant region here and a variable region. That's what we're talking about here. That's what changes. And 2 light chain here with the variable region. So what we're talking about here is the steps happening between the formation of a precursor B cells from a progenitor B cells, where the heavy chain, so the variable part here, will be changed. And the second step, we're talking about how this variable part in the light chain is formed when the precursor B cell mature into an immature B cell, which then will move to the lymph nodes. So each B cells has 3 basically genes here. Has 1 gene that gives origin to the heavy chain, this one right here, and 2 genes that give origin to the light chain there, okay? Now, if you look at the structure of these genes, they have an amazing thing, which is they have a part of the gene which gives origin to the constant part, this one right here. This is the same for all the B cell receptor and therefore for all antibodies. But then you have another region there, which is referred to 3 different regions: the V variable region, the D variable region, and the J variable region. This is what will give origin to the variable region, both in the heavy chain and the light chain. How is the variability originated? Is by the fact that in reality, this V area, the area consists of different parts, with one of them that ultimately will stay in the final gene that produced this variable region. So here you have about 40 possible V segments present here, another 23 possible D segments, another 6 possible J segments here. And so now the difference between the heavy chain and the light chain is that the variability in the heavy chain is generated by all the basically random assortment of region from these 3 parts, whereas in the light chain, the D region is missing. Okay, so you have only the variable segments here and the joining segments here that are present that can be causing the diversity. All right, so what happens is that now in these steps here, in the heavy chain arrangement, you have that this gene actually undergo recombination. So to start with, all B cells will have exactly the same gene here. But now you have recombination. So basically what you have is that this gene will lose some genetic material and basically will form a final gene which comes from the combination of different regions, let's say V2 with D22 with J5 here, right? So, and this, it's happening in this step right here. What you have is that this combination of different parts is caused here by recombination. So the genetic material here in that gene is changed. In all different cells, that event is different. So basically causing that these B cells can become many different types of precursor B cells with different genes for that heavy or variable part of the heavy chain, right? And now the same thing happens for the light chain except for the diversity region when the precursor B cells gets into the B cells here. So by doing that, you can produce basically starting from the same B cells here. You can produce so many different type of B cells with all possible combinations here. Okay, so possible combination of the V segments with the D segment and the J segment, which ultimately leads to different genes in different B cells, and therefore different B cell receptors, and therefore different antibodies. The antibodies produced are basically the B cell receptors without the transmembrane domain in different B cells. That's how all that diversity is generated. That's a first amazing step in which all the diversity, so all the different B cells, and their T cells is the same thing, can be produced to recognize so many different type of antigen, even though these antigens never enter the organism. So then you have all these different cells that can react against all these possible different antigen. And so the cells with the specific B cell receptors gets activated when the antigen comes out. Just by chance, you have one antigen that matches that B cells will get expanded, all right, among this big variability. This is the first level, so we can move on here. So, and these will lead basically to the different formation of the variable region of both the heavy chain and the light chain here, even though in the light chain there is no D segments present, all right. Now, the second level is actually what happens now after this. So going back, so now this is where we are at. So here you have all these B cells, which then will move to the lymph nodes. Okay, they're all in the lymph nodes, and that's where they're getting ready basically to be presented the antigen and to expand and form the plasma cells and the memory cells if the antigen shows up. However, there's another step happening there. Once one of these cells, okay, gets stimulated to become a mature B cell. So once the helper T cell presents the antigen to any of these B cells now you have a second level of variability which is introduced by what is called somatic hypermutation there. So you have this B cell receptor is already fairly specific for that antigen. Okay, so it was already, let's say, is already good enough to react against that. But you want to produce B cells that have an even better receptor, okay, that is so amazingly has a high affinity that is going to remove these antigens so quickly and therefore the pathogens so quickly. And so what happens in the lymph node is that when the B cells start expanding after this level here, the presentation of the antigen, they start expanding. When they start expanding, they actually go through a series of mutation, random mutation mediated by an enzyme, which is called activation-induced deaminase. So the activation of the B cells here activates also these deaminase, which start producing a series of point mutation. So now these cells are expanding into clones, which will have some slight differences in the B cell receptor among each other. They're all fairly specific, probably to the antigen, but some will be at this point a little bit more, just by chance will become a little bit more specific, some other a little bit less specific. Together with that, now you have a selective mechanism there. So the B cell has been activated by the antigen, is producing many B cells that are hypermutating. So these cells have slight differences in the receptors there. And now they get somehow tested within the lymph node. This will be a lymph node. And those that have lower affinity, they're actually forced into apoptosis to die. Whereas those that have a higher affinity, they get stimulated by further exposure to the antigen and to expand. These are the ones that do ultimately expand into the plasma cells and the memory cells. So by doing that, basically what you have is that even the naive B cells, which now is in the lymph nodes, it gets even higher affinity from the process of stimulation at the beginning to the end, as acquired by producing many cells through somatic hypermutation, has produced basically some cells that have even higher affinity, which are the ones that ultimately will turn into plasma cells fighting against that antigen and the bacteria that carries it, and some of them will become memory cell. So two mechanisms that causes that variability. The first one is the genetic recombination, which happen at the gene level through the maturation process of the precursors of the B cells ultimately. And then after stimulation, you have somatic hypermutation happening, which ultimately leads to the formation of even more powerful, if you want, type of B cells with an affinity for the antigen even much higher than the starting one. All right, that's all we're going to discuss here. So we're done. That's the end of the semester here. I'll take questions through any of our meetings if you have any. All right.